All right, thank you. As Miranda mentioned, my name is Phillip Lujan, and I'm one of my roles is the plant diagnostician here at NMSU in Extension. So I guess moving straight to it, I broke this presentation down into two sections. The first one is kind of just a small general plant pathology refresher. And then the second part is the more fun part. It's the fruit tree disease example part. All right, so getting started, so I want to go over some key plant pathology concepts that everybody just should get familiar with. The first one is that there are many different living, which we call biotic, as well as non-living or abiotic entities that can have a negative effect on plants. The second one, and living here in New Mexico, where we receive very little to no water. The second one is plants that are under stress, whether it be biotic or most likely abiotic or non-living again. Those are going to be more susceptible to diseases than plants that are growing strong. And then third is successful disease management. That depends on first accurate identification of what's causing the problem, and then how we can use some of the available control methods in an integrated manner. All right, so next we're going to go into some definitions. Some of these, plant disorder and plant disease, sometimes they're used interchangeably, but really a plant disorder is just any abnormal growth or development in a plant or fruit. Here, an example here in this upper part, this apple here, this is a bitter pit, and that's due to a calcium deficiency. Now, the difference between a plant disease and a plant disorder is a plant disease is any abnormal growth or development in a plant. So very similar to that disorder part, but this one is particularly caused by an infectious microorganism or a pathogen. So that's the two differences. The first one, disorder, can be kind of a more general term, but a plant disease is more a term for an infectious microorganism. Now, what is a pathogen? So a pathogen is derived from the Greek words to give rise to suffering. So really a pathogen is just any agent which causes diseases in plants. And that can be bacteria, that can be fungal, viral, even insects and nematodes as well. So again, back to this basic concept in plant pathology between those living and non-living entities that can have a negative effect on plants. So some of those, as I mentioned before, some of those biotic agents can be fungi, bacteria, viruses and viroids, parasitic higher plants. Those, for example, are mistletoe or dodder, kind of that yellow stringy stuff sometimes you see on the side of the road after it rained. Again, those biotic disease agents are called pathogens. However, not all microorganisms cause diseases. There can be good ones, there can be bad ones, and many of these are necessary and beneficial to plants and or animals. And I'll give some of those beneficial examples later on in the slides. Now moving on to abiotic or non-living entities that can have an effect on your plants. That can be temperature extremes. This is especially evident, all around Las Cruces you can see, and probably all around New Mexico, we're just having some really crazy weather patterns. Some days it's really cold and it gets below freezing. The next week it's 90 degrees. So just that fluctuation does have a lot of negative effect on your plants or trees. Moisture extremes. Here again in New Mexico, we don't have much rain or water events. So most likely those moisture extremes are going to be due to drought. Other times, more in an individual home, you can overwater your trees as well, but that's very infrequently. Nutrient extremes, same thing. You're adding too much fertilizer. There can be some nutrient toxicity. Soil problems, especially here. Again, we have very alkaline soils that can cause issues. You can see salt damage. If you plant a tree and the soil's too compact or on a hard pan, maybe those roots aren't able to spread out as they normally could, and that can affect your tree growth as well. Pesticide toxicity is a big one. Again, you need to first identify your problem before you start, spraying things and causing more problems. So that's a big one. And then finally, improper cultural practices. So that can either be, planting your tree too deeply. Those roots aren't able to get enough oxygen and grow properly, or even planting too shallow, and with our crazy winds, it's just going to knock it over. Or planting the wrong tree at the wrong place. That can also have an effect on how your tree grows. So now a little bit of what we do here in identifying the causal agent. If it's the first thing we need to do is determine if it's abiotic or environmental problems, or if it's an actual pathogen or biotic problems. So some of these environmental issues, oftentimes you see one or more plant species that are affected. So, different types of plants that are affected. Sometimes from one day to the next, your tree dies. That's more likely due to environmental problems more than a pathogen who wants to keep that tree alive and just kind of continuously feed off of that. The symptoms really do not spread. So it's there. They're affected, but they're not going to move down the row from one tree to the next. So it's a localized issue. And then other times the distribution pattern is typically uniform. So the whole row, the whole orchard, maybe a block from an orchard, that's kind of a uniformity is a big telltale sign of that it's an environmental issue. Now, for a pathogen, typically only one plant species are affected. And then the symptoms typically develop pretty slowly. So you're going to see that develop over time. It's not going to be very sudden versus the environmental or cultural problems. And then the symptoms can spread from plant to plant. So if you see one tree starting to wilt, not doing well, and then it can be spread either through insects, through your tools, or through the roots touching each other. And then that next tree will start to show some symptoms later on down the line. And then the distribution is typically kind of clustered or very sporadic. It's not very uniform as we see with the environmental problems. Now, moving on to the plant disease triangle. So when a tree or a plant gets a disease, you need to have three things all work together at the same time. You need to have a virulent pathogen. So the fungi, bacteria, anything, it needs to actually cause infection. then you need to have a susceptible host. If you have a pathogen that's able to infect, but that host is resistant, you're not going to get disease. So it needs to be a susceptible host to that pathogen. And then finally, once those two are playing together, then that favorable environment comes into play. And that favorable environment is usually moist temperatures, humid, high humidity levels, usually after rainfall. All three of those things need to come together, and then the plant will get a disease. So next, a little bit more on definitions of symptoms. Symptoms are really the response of the plant to an attack by a pathogen or to any adverse conditions caused by an abiotic disorder. See, for us, we can tell the doctor, what's wrong, what we're feeling, what's hurting. For plants, they can show symptoms and kind of lead us, point us in the right direction as to what may be happening. And some of those examples are wilting, so just not getting enough water, not holding turgor, the leaves start kind of drooping. You can get yellowing of leaves. You can get leaf spots, stunting. The tree or plant is just not growing as well as it should. You can get fruit rot, even root rot. So all of those are symptoms. However, those are not really specific to the causal agent. So the symptoms of wilting, it can be due to maybe lack of water. It can be due to a pathogen. So just because we see a wilting plant, that doesn't really clue us in as to what exactly is the problem. Now, signs are the actual visual presence of the pathogen on the plant. And some of those examples are rust, which you can see here on this apple. Pycnidia, those just house spores, and they can be released through wind or rain. You can see ooze here, so this is a bacteria. And then you can see powdery mildew, so that's another fungal issue. But these are the actual visual presence of the pathogen. Now, to make our life more complicated, there are organisms associated with disease plants. So first is that primary organism. That's the organism or the pathogen that is actually directly responsible for the disease. After that happens, then you get your secondary organism starting to move in, taking advantage of that weakened plant or tree, of that tissue. So these are usually relatively weak pathogens. They need that primary organism to start affecting that tree first, and then these things start moving in. And then finally, you get your saprophytes. These are really, they don't really cause any issues on the host themselves. But once that tree or plant is pretty much dead, they start feeding on that decaying organic matter. And so here's an example here of a redbud tree. It's pretty much dead. And then you start to see some of the saprophytic growth. Now moving on to some plant disease management methods. There's about 5 of them, and I'll go into each of these in some small detail. However, successful disease management, it usually depends on, the integrated use of all of these things all together. And that's IPM, Integrated Pest Management. And the best management tool is prevention. So like for us, eating healthy, exercising, taking vitamins, you want to, prevent any problems. And the same thing goes for your trees or your plants. If you keep them healthy, well watered, well taken care of, they usually do a great job in preventing diseases themselves. So the first one is genetic resistance. That's usually the most effective means of control when it's available. Sometimes it's not, but there are cases where genetic resistance is a really good control method. However, this must be continually monitored because these pathogens are, pretty adaptable, pretty smart. They will start to develop virulence to those tolerant plant materials. So they'll start to genetically modify themselves in order to eventually infect those what was once resistant plants or trees, and they'll start to, infect them. The second one is cultural management. So that starts with either, choosing the right species or cultivar selection, sanitation practices. That's key. If you're going to a nursery, you want to inspect what you're bringing into your area or your orchard, your garden. So the use of disease-free planting material is also key. Then you want to choose a proper site selection, planting time and planting techniques. Again, water and fertilizer management are important. Insects and weed management. There's a lot of weeds that host pathogens as well as insect vectors. So if you get rid of your weeds around your orchards or your trees, then you can kind of limit that spread. And then crop rotation. Again, these are more for annuals. It's pretty difficult for fruit trees to be continually rotating them. Next is biological management. That means just the management of pathogens by other microorganisms. So this is constantly occurring in nature. There's a lot of native strains of microorganisms in our soils that helps our trees and plants fight off some of these pathogens. And here's an example here. This is a trichoderma. This is found universally in soils. It's a beneficial fungus. And what they did is they tested, this is botrytis, so it's a gray mold. It grows on strawberries a lot, different types of fruits. And what it does is this trichoderma just grows so quickly versus the actual pathogen that it starts by day 3 overtaking this pathogen, and it can it's really not very useful anymore. So that biological management is really neat. Another one is, here's a plant parasitic nematode, and a beneficial, again, fungus found in the soil. It produces these circular structures that when the nematode goes through them, it senses that and it starts closing off, and it traps those nematodes there. So they're unable to, feed off of any of their host plants, and they eventually starve and die. Finally, if all else fails, chemical management. Some examples are fungicides, bactericides, nematicides. However, if you apply these chemicals, you need to make sure they're not, less toxic to the plant than to the organisms they're designed to control. So if you're spraying and it's affecting your plants or your trees, but it's not really controlling your organisms, that's not very useful. And then a lot of these fungicides that are out there are really fungostats, meaning they kind of just temporarily stop the growth of these fungi. It doesn't really remove them or kill them. It just renders them unable to grow for a short time period. All right, so now we're done with that small review. Then we'll go into straight into this fruit tree diseases, and I just kind of listed the ones that we see most often, and then we'll work from there. The first one, apple diseases. A big one here is fire blight to look out for. It's caused by a bacteria called Erwinia amblivora. It also infects plants that are in the Rosaceae family. So any of your apples, cherries, roses, peaches, pears, and berries, they can all be affected by this disease. This bacteria overwinters or survives over the winter in previous year's cankers. So a canker here is just this blackened area and in these boxes here. So this is where the actively growing bacteria is in that branch. They can also survive in healthy buds. They're mostly spread by contaminated pruning tools. So if you're pruning your trees and they're affected by this and you don't realize it, they can survive on some of these pruning tools, and you can just be spreading it that way. Insects and bees can also spread through when they're feeding. So some of the initial symptoms that you can see, the infected twigs become water soaked, dark, sunken, and dry. So again, these lesions here. There's some oftentimes cracks can develop in the branches. And then kind of a tall tale sign that it's fire blight is these young twigs and branches. They start dying from the terminal end, and then they start, they look just like they're been burnt, and then they start bending. So that's what we call a shepherd's crook. You can see that right here. Here are more examples of lesions as well as the shepherd's crook that you can see. So if you see this on your apples or any of your rosaceaes, most often the case that it is fire blight. Now, what do you do when you do suspect fire blight? Again, you want to start, I'm going to say this often, start with disease-free planting material. If you do see those lesions, you want to prune out those infected branches at least 6 inches below the lowest lesion you see on that branch. Again, when you do that, though, you want to clean those pruning tools by dipping them in either rubbing alcohol that you can find at your local drugstore, 10% bleach solution. So 10% bleach, 90% water, mix that. Then you can dip your tools in there. Flame sterilize, you could, but you don't want to start fires. We're dealing enough with those already. So there are some protective sprays. There are copper fungicides, antibiotics. Those are usually applied during flowering, but they're pretty expensive. They're really only cost effective for those commercial orchards. And if you do suspect fire blight, you can send a sample to us. You can, chop off that shepherd's crook, send those lesions to us on the branch, place it in a paper bag, and then send as soon as you can. Now another, this one's not so common here in New Mexico, but it does black pox. It affects apples. It's caused by a fungus called Helminthosporium. It infects apples and pears. It's really considered to be a minor economic importance. It can affect the branch fruit, which you can see right here, as well as the leaves. Again, this one also overwinters in those old bark lesions. So you can see some of these lesions right in there. Now, when it's about 80 to 82 degrees, that's the optimal growth temperature for this fungi. And again, some of those symptoms that you can see on the fruit, pretty obvious. You see the conical, smooth, shiny black swellings here on the new growth. And then once those age, they become kind of this really dark black looking spotting thing. On the leaves, they're small circular green spots, oftentimes with a red halo around it. And I think, well, here's some more pictures of black pox. So these are on the branches, the fruit here. If you do suspect it, again, use disease-free planting stock to avoid it. Selective pruning. So that microclimate, if there's a lot of branches together with a lot of foliage, it can create a very humid environment. So you can selectively prune out some branches to let that airflow go in and kind of reduce that humidity around there. And then you want to prune out your infected tissue. So, and if you see those lesions, again, prune that out, and then sterilize your tools as well. There are fungicides available if necessary, usually not very necessary. And again, we don't see this very often. So if you do see it, send some symptomatic fruit or branch pieces to us and we can identify it. Now next, apricot diseases. A common one is Botryopsphera canker. It's a fungus. It has a wide host range from apples to cherries, even to oaks, ashes, all the way to your evergreens, as well as grape. These fungal organisms, they kind of hang around until you get a fresh pruning wound, either in the spring or summer. And then usually after a rain or an irrigation event, when that, that you get that microclimate, that humidity going, that moisture, then these start to infect your trees. They overwinter in previously infected wood, and you can see some of that here and here. So some of those symptoms, like this top picture is gammosis. You can just see this gummy amber colored ooze coming from the bark. You can get limb dieback, and then the xylem tissue and the cambium are discolored brown, which you can see right here. And here are just a lot of pictures of Botryopsphera canker. You can actually see the cankers here. Once you do a cross section, you can see the infected tissue, more of that amber gammosis, and then you can get branch dieback as well. So you want to remove any of those infected limbs at least 12 inches below any observable canker or symptom. You want to prune after harvest and try to, prune before any rainfall events that you can see. You can treat some of these pruning cuts or wounds because again, this Botryopsphera usually needs, a fresh wound to start infecting. You can use paint or sealants. It's less effective. It's less effective if you have a large amount of trees, just because of a lot of effort and attention is needed. But if you have a few trees here and there in your backyard, you can do this, and it tends to do well. Fungicides are also available to treat some of these wounds after pruning. An example of that is myclobutanol. And then if you do see some of these cankered limbs or dieback, you can send us some of those and we can try to look for Botryopsphera. Now, apricot diseases. This one's a pretty neat one, at least to a plant pathologist, maybe not to the grower themselves, but crown gall. This one's caused by a bacterium called Agrobacterium tumefaciens. You can find this on apple, cherry, pear, plum, apricot, as well as roses and willows. This bacteria survives in the soil as well as that in the gall tissue. So you can see here, these blue circles, here's some of that gall, and then here's an extreme version of that. Again, this one as well, it can only enter a tree through wounds or through wounded roots, whether that be during planting, grafting, insect damage can cause problems, or if you're mowing, weed whacking near the crown there and you nick it, then that can be enough damage for that weak pathogen to enter and start causing problems. It is most damaging to young trees. Again, they need more, if they start affecting it at the crown, they don't have enough roots to kind of overcome that. So some of these older trees can survive a few galls here and there. Again, those galls, depending where they're at, they can interrupt the flow of nutrients and water. And then signs and symptoms, they're very observable. Just these irregular rough galls, either on the roots or on the trunk. Young trees can be stunted. Again, they don't have enough nutrients and water to grow fully. And then the older trees can develop secondary wood rots, just because they're not moving that water up to the foliage. Other examples here of crown gall. Just see a bunch of galls forming kind of all around. Crown gall management. You want to handle with care your plants or trees, especially when transplanting, to avoid injury. Again, during planting or throughout the life of the tree, just be very careful when you're working around that, not to nick the trunk or the crown, because again, this bacteria does need some help moving in. There are some pre-plant biological dips available. This is an Agrobacterium radiobacter. So even though it has the same genus name, species is different, and this is a good guy, a good bacteria. If you take out a tree that was previously infected with crown gall, just try to remove most of those old tree roots, or even better is just plant away from that area. That soil will still have that, that bacteria still in that soil. So if you notice crown gall developing, if on a recent tree, you want to dig up that tree as well as the soil and just dispose of it. For established trees, again, as I mentioned, they can tolerate some amount of infection depending on the location. If it's right on the crown, maybe not so much. If it's on a root, then maybe they can overcome that. So for a sample, really all we need to see is like a really detailed picture. So you can send that through email, or you can even send a piece of the gall, and we can look at it here in the clinic. All right, so now moving to cherry diseases. Bacterial canker is one that we see often. It's also called gamosis or soursap. This is caused by a bacteria called Pseudomonas syringae. It hosts, a variety, almost all the stone fruit trees, apples and pears can also be infected. This bacteria can infect branches, twigs, buds, leaves, as well as fruit. So pretty much all of it except the roots. The bacteria overwinters, again, in those cankers, which you can see on this bottom picture here. or on in infected buds. So in the spring, especially after the rainfall, these bacteria just multiply like crazy, and they start oozing out from some of these cankers. And you can see that on this top picture. They don't like so much, our 100-degree temperatures. So they like that, 75 to 85-degree temperatures. That's when you start seeing a bunch of this occurring. Once it reaches above 85, usually the disease development stops. It's just too hot for them to grow anymore. Some of the symptoms, as I mentioned, cankers and dying branches due to the girdling. So if you see this type of canker here, it's just limiting water and nutrients above. So you're going to start to see some dieback above those cankers. On twigs, again, those cankers are areas near the base of the buds. You can see gumming, and then leaves and shoots can be killed during dormancy. Just more examples of bacterial canker. Again, in the foliage, the fruit, branches. Bacterial canker management. The disease is more severe on cold injured trees and on sites with poor soil drainage. You want to, again, disinfect your pruning tools. That bacteria can be transmitted through these. It's just good standard cultural practices to clean your tools, especially moving from one tree to the next. Pruning is best after harvest during the summer when the conditions are hot and dry. Again, the bacteria doesn't really like too hot of a temperature, so this is a good time to prune. Symptomatic limbs should be pruned out as soon as you can when you first see it, several inches below that canker. There are copper applications that can be applied. It's not very effective for this bacteria. It's how it goes, I guess, at times. You can send cankered limbs or symptomatic leaves to us, and we can try to ID this bacteria. Bacterial spot on peaches. This is caused by Xanthomonas campestris. Again, this bacteria can infect nectarines, apricots, and plums. It can overwinter in those dark, gummy branch tips, and then as well as diseased twigs. This bacteria does like a little bit of a hot temperature, but it does need that moisture. And then sometimes when it's windy and sandy, it likes that because it can, that wind and sand kind of, scars some of those fruits, and it's able just to penetrate those fruits and leaves easier. Symptoms on leaves are generally not seen until after bloom. And then a few weeks before harvest, the fruit becomes more susceptible. Again, with that wind and the sand, those abrasions and nicks on the foliage and the fruit, it just makes it much easier for that bacteria to get in there and start infecting. Some of the symptoms that you can see, like on this picture here, are fruit spots. You can see twig cankers. You can see leaf spots. On the fruit, sometimes it cracks. There's gumming, and then the water-soaked tissue as well. And here are just more examples of bacterial spot. There are some varietal resistance in these peaches for bacterial spot. You can establish a good cultural practice is establishing sod strips or even gravel to kind of suppress that blowing dust in our wind from our winds. Again, they need that those abrasions to infect. So if you put some sod strips or wood chips, something like that, to limit those abrasions, that would be a good cultural practice. There are some bacillus-based biologicals available for management of bacterial spot. And again, there are also some copper-based bactericides available, but from what I've read, they're not that effective. So take it as you will, I guess. You could send some symptomatic fruit or leaves to us, and we will try to determine if it's bacterial spot. Now moving on to brown rot. This one's caused by a fungus called Monolinea fruticola. This is a pretty destructive disease of stone fruit. You can find it in nectarines, apricots, plums, and cherries. This fungus, it overwinters in mummified, or what we call mummies. It's just mummified fruit. So you can see in this bottom picture here, this is just the fungus and all of its spores just hanging out there, waiting to infect another fruit or twig. The spores from the fruit from here, they land on those wet blossoms or new shoots, and then they can cause reinfection. The symptoms of this, they first appear in the spring. Some of the symptomology for brown rot is blossom and twig blight. You can see cankers, and then the fruit rots. The flowers can wilt, they turn brown, but they do remain on the tree. Fruit infections, so if you fill the fruit, they appear soft, they have brown spots on them. And then again, those mummified fruit is a telltale sign for brown rot. You can see here on the flowers, the fruits, and here's another mummy. So these fungal spores are everywhere. So any type of injury to a ripening fruit, it'll provide a good entry point, whether it be insect damage, hail, even if a twig starts rubbing on it, or any mechanical damage, it just allows that fungus to enter and cause trouble. Good sanitation practices are key. You want to remove any of those mummies that are on there, and then you can prune out cankers in the late winter during dormant season. There are few peach-resistant varieties available, and there are fungicides available, such as chlorothianol, Captain, or myclobutanol. If you want to send a sample to us, send those cankered limbs or those symptomatic fruit. Next is nectarine diseases. One is shot hole, it's called, or corneum blight. This is caused by a fungus called Wilsonomyces carpophilus. There's also another name that I forgot what it was. I should list that. This survives on infected twigs and buds. The spores are produced throughout winter, and they're just spread by rain or wind. This disease does like prolonged leaf wetness, like in the fall or midwinter. For some symptoms on these twigs, you can see in this upper picture, small purplish black spots. They start enlarging and then turning brown. And then on the fruit and leaves themselves, you get these small spots that are purple, then they turn brown, and then they fall out, and then they look like somebody, put a shotgun to it, and you see a bunch of these shot holes here. Here's better examples on the foliage and on the fruit. For shot hole management, primarily managed with fungicides, Bordeaux mixture. So that's copper sulfate, lime, and water. And you can spray that. You can prune out and infect and destroy any infected wood. So if those trees are sprinkler irrigated, you want to angle those sprinkler heads low enough from wetting the canopy and just, shooting those spores everywhere. You can send symptomatic leaves or twigs to us for identification. Next are plum diseases. Black knot is one. We don't see this too often, but I wanted to mention it. This is caused by a fungi called Apiosporina. Really, its hosts are cherry and plum. The disease is present only on the woody parts of the tree, so usually you find these on twigs and then branches. The symptoms become pretty visible in the late summer or on the next season on the new growth. The fungus itself overwinters in these knots on those twigs and branches. And then this one does need some rainfall to cause infection. That's maybe why we don't see it that often. So signs and symptoms, you can easily see these knots here and know that it's black knot. And here's more black knot. Black knot management. So new plantings should not be made next to the old ones where black knot is occurring. You can, if you do find black knot, you want to remove and prune out any of those infected branches, at least 4 to 6 inches below. On large branches and trunks, those knots can just be cut out themselves. Once you do remove those knots, fungicides are available. You could spray. The spray should be applied in early spring when the flower buds are beginning to open, just to protect those green shoots. Again, if you want to send us a picture of the knot itself, we can ID it through email usually. All right, so we kind of blasted through all of those diseases and all the problems that could happen, but just remember there's a lot of living and non-living problems or entities that can cause a negative effect on plants. The plants that are under stress, either due to watering, due to physical damage, insects, anything like that, those are the ones that are going to be more prone to those diseases. So just take a good look at your trees, your plants, make sure they're well fed, they have enough fertilizer, nutrients, enough water, they're pruned well to get those microclimates addressed, and then you'll have usually a pretty healthy tree. Just remember again that plant disease triangle. You may have the pathogen present, you may have a susceptible host, but one thing we can control usually is the environment that they're in. Sometimes we can put soil amendments to make that soil better. Sometimes through pruning, we can reduce the humidity. We can increase the water, or in other cases, decrease the amount of water present. So there are things that we can address here so diseases won't occur. And then finally, successful disease management. Again, we're here to help you to accurately identify the causal agent. And then it's up to everybody for the integrated use of those available control methods. And that's what the NMSU Plant Diagnostic Clinic is here for. Probably Dr. Beck from last week's probably mentioned this, but we are an integrated plant diagnostic clinic. And what that means is, we can look at biotic issues caused by, diseases and pathogens. We can look at abiotic issues, make that determination between, is it an actual disease or is it more of an environmental or cultural issue? We do take in insects and arthropods for identification, and then we take in plants and weeds for identification as well. We are permitted to receive unknowns. So any of these plant materials from within the contiguous 48 states, we're not allowed to take anything from Alaska or Hawaii. For more information about what, sampling, how to sample, sampling techniques, sample forms, or where to contact us, go to plantclinic.nmsu.edu. Now, I just a quick mention about sample submission. So that's important. That's very important. So the samples must contain the right material, right? So if you suspect that, there's a root issue, we need to see the roots. We can't see the branches and make that determination. Or if you see leaf spots, send us a good amount of leaves, and that helps us with our testing. So just make sure, again, diseases may show up on any part of the plant or tree. Just look, take a good look at it and send the areas that you suspect are problematic. And then if you visit that website, we do have the plant specimen submission forms that you can fill out. And usually you send those samples through your local extension agents. That's our preferred method. You go visit with them, or you can, email them, contact them, and they can possibly go out and visit. Just get to know your extension agent. And if you do that, the extension agent knows how to, quickly send those samples to us, and everything is free. All right, so thank you. Here's my contact information, office phone number, email is here. That's a preferred method because I'm usually running around. So I answer email, pretty quickly. So feel free to email me, and I will, I guess, take any questions.
Thank you so much, Phillip. That was a lot of information, very good information to have too. One question we had was, what is considered a young tree?
Oh, that's a good question. I guess it's in the eye of the beholder, but usually 1, 2, 3 years. Okay. That's pretty young to me. So it could be based on, somebody else's opinion as well, but not very established.
Okay. It doesn't have anything to do with like if it's bearing fruit yet or not?
No.
Okay. Also, you mentioned pruning out diseased branches a few times and the importance of sterilizing the tools, but is there a best time of year to prune?
Yeah, so it usually depends, I guess, on the disease you're looking after. So during the time when those diseases aren't very active, usually it's harder because you start seeing symptomatic tissue happening in the spring. So early spring, if you can, or before, way before rain events occur, that would be a good time, or in the late fall.
Okay. I suppose it's a balance too, because then you have that damaged site that other pathogens could be introduced.
Yes.
And then just kind of like a behind the scenes, like how are you identifying the diseases? Is there a test, or is it through microscope?
Yeah, there's a whole lot of things that we do. We first look at the sample under a microscope, try to make that determination between, environment, or is it an actual problem. Then we kind of look at the disease margin. So it could be maybe a blackened area versus a healthy tissue. Then we can plate that out on, like a sugar and potato and gelatin mixture on a petri dish. The, pathogens like to feed off of that. So they start growing out of that diseased tissue, start growing out to the plate. And then from there, we can look again at the microscope, compare references, or we can even go further into doing molecular DNA extractions and sequencing and make that determination as well.
Oh, wow.
Yeah, there's a lot of tests. It's fun.
So that's why you study it for so long.
Yes.
And then another question came in. What do you do with the disease clipping that you pruned off?
Yeah, so usually put it in a black trash bag and throw it away. That's probably the preferred method. Other methods that people list are burning, but we're dry here and windy, so that may not be a good one to do. So usually just rake it up, put it in a trash bag, close it tightly, and just dispose of it that way.
Okay. So I guess key is to make sure you're getting it away from your trees.
Yes.
Well, that's it. Thank you so much, Philip. Thank you.